One. And we are recording episode 1003 on Tuesday, November 29th, 2022 at 2:06 p.m. Eastern time. And Dr. Bhargava just gave me a lesson on how to pronounce her name. And I know I'm going to mess it up, but I'll try. It's so tongue to palate. So Dr. Aditi Bhargava. Aditi. Did I get it
Yeah,
right?
you got it.
Anywhere
You got it.
close? All right.
Yes.
Well, from
Thank
here
you
on
for
out,
trying.
I'll call you Dr. Bhargava from here on out. But for all the new listeners, you first came on about a year ago, and then you came on again with Dr. Malone, and then you came on again with Martha Fowler and discussed all things mRNA, the lipid nanoparticles, the uneven distribution of the mRNA target package. I don't know if that's the correct terminology, and how that uneven in distribution. And I believe you went through a PowerPoint slide your first time on here and showed that at the time, hypothesizing that that could be responsible for the seemingly drastic differences in effects people have. It's not that some people kind of got myocarditis and some people get one shot and three boosters and they're fine. Some people get a shot and they're dead that evening. And so there has to be an explanation for why it is so drastically different. And your hypothesis, if I'm recalling correctly, is uneven distribution or allocation of the actual the good stuff, the package, the mRNA. And now that we're seeing, I've had on Dr. McCullough and Sasha Latipova and Dr. Fareed, and they've all been talking about what is called, in quotes, the hot lot, capital H, capital L hypothesis. And it's actually credit to my mom. I sent it to her, and she responded, "You need to have on Dr. Bhargava about this." And I was like, "You know my podcast better than me, so shout out, mom." But Dr. Bhargava, for all the new listeners, could you first introduce yourself, and then we'll jump into the podcast.
Um, Thank you, Tommy, for inviting me again. And so I am a molecular biologist, and I have been at University of California, San Francisco for over 26 years. And I am a professor, and actually now Professor Emeritus, officially sort of retired, and have started my own own thing,
Okay.
just because there is a big need, and as we can see, there's a big need in terms of even scientists don't know how to analyze data, and so we're working on some software that can analyze big data, such as, when you sequence huge amounts of genomic transcripts or proteins, or called omics. So anyway, so that's what I'm going to be doing. But since I'm emeritus and I've worked or earned it, I still get to keep my research endeavor at the university. I just don't have to worry about raising or writing grants day after day and worry about where my salary is going to come from. So I have this freedom. And of course, as you know, I've been doing that COVID study on trying to understand how COVID impacts our health, as well as whether the vaccines are actually effective in real world, and whether or not vaccines induce or associated adverse events are more frequent than we think. And it all stems from my interest in mRNA therapeutics. As you know, that UCSF had filed a patent on my behalf for mRNA or RNA therapeutics, not just mRNA. There are other kinds of RNA interference as well as, and it was using also for a delivery platform, which was using specific kinds of encapsulation techniques, not so much as a lipid nanoparticle, which is what's being used here. So that's what initially had sparked my interest in this technology, and very excited about it. And as I read more and more about it, I became highly concerned. And so hence my hesitation in adopting this technology for vaccines in such at such mass population level in healthy people.
And that's what I think the first thing I ever saw of you. I believe it was on, I think it was Aubrey Marcus, and I'd seen a clip on YouTube of you discussing just that, and it was just the most measured, just concern. It wasn't some hysterical, this is the end of the world. It was just concern. And whenever healthy and well-founded concern is immediately attacked and criticized, that's kind of when the hairs on my neck go up, and it just seems like something's not right. Like you should always, it's always, guy just moved into an apartment. I'm on the 5th floor. I've never been higher than the 2nd floor. First thing I did was I just found the emergency exit. And it's right next to my apartment. I was like, cool. But if I had started looking for the emergency exit and all of a sudden, like, employees of the building came up and they're like, what's the matter with you? Why are you looking for the emergency exit? You don't trust it? I'd be like, no, I'm fine with it. It's just, I am about 60 feet up. If I jump out of my window, I will die. So I would rather find the emergency exit in case of a fire. No one in their right mind would criticize that. That's kind of where my intrigue with all of this comes, is it's a very, I don't know what the answer is, and I don't know why people are so hesitant of criticism, but that's kind of where I come from. And it seems, because everything is obviously still a hypothesis, or technically all sciences are still a hypothesis, but it seems like there might be an explanation, or at least a coalescing of disparate facts that are coming together and maybe justifying your initial concern, and what boils down to kind of the uneven distribution of the mRNA material in the shots. Is that anywhere close to correct?
Yes. I mean, you're right about it's it's a conundrum as to why suddenly scientific critique or dissent is being taken as though we are anti-science or anti-vaxxers or whatever, So vaccine has become sacrosanct, right? It's a cult, it's a religion now. It's not a treatment or a drug anymore. It's like if you either believe in it or you don't believe in it. That's not science.
No.
I mean, right?
No.
If you ask a scientist even about, I don't want to go into where the existence of God, but if somebody cannot debate that, then you would call that non-scientific, right? So if you cannot debate it, then how do you call it science? And like I've said before, vaccines themselves have, in the past, there have been instances with vaccines, they've been recalled, they've been refined, they have been better tuned, and there is no doubt that there are some vaccines that have definitely helped mankind. Just like antibiotics, they've really helped our survival, and they've been one of the most important discoveries. But overuse of antibiotics has problems, and for the longest time, the medical community didn't think about overprescribing, and now we know what's the problem. So anything that you overuse, you're going to have problems. So why not with vaccines? Why you keep and in any case, vaccines are, there is we'll stick to the mRNA vaccine. So today, what I will attempt to do maybe is to just go back um, and give a little bit more background, and so for people can understand. So there's obviously, when you talk about these hot lots, I don't know, I haven't used that term, but there are definitely concerns that are manufacturing concerns that are that exist and that have been raised. When you scale up technology, that's always an issue. So one of the biggest problems with this mRNA technology is that it had never been scaled up, right? It's the first time it's being used in such so many people in so many millions of doses. Before that, for over a decade, they were only being done as clinical trials, phase 1, phase 2, 100 people, whatever. So when you make these things in small batches in a very controlled environment, that's very different than when you start scaling up, you're setting up manufacturing units during a shutdown when you don't have, right? So this was during the shutdown, right?
Yeah.
Because the emergency use authorization happened during the shutdown. So at that point, China shut down, which is the most used country in terms of manufacturing and things like that. All the raw materials, how do you move them around? There is the shipping logistics of all of that, right? So you're setting up something that has never been tried in a time where you have all these other logistic issues to deal with, and yet they are able to set it up in numerous places, numerous countries, obtain all this raw material from magically from wherever. So those are things that anybody who is not who wants to think should think, right? I mean, when you are if I cook at home, and that's great, but if I were to set up a restaurant tomorrow without any experience, to scale it up to feed maybe 100 people a day at different times coming in, it's maybe I'm setting up myself for a failure. Maybe I will succeed, maybe I'll not. For me to be able to then reproduce the same kind of cooking that I do for 5 people instead of that for 500 people day after day without any experience, it is a little bit hard to imagine. Without any, what kind of raw materials, how many raw, do I need? What kind of cooking utensils do I need? How much of spices do I need? What's the combination? None of that I've tried, and I'm going to just do it, and I'm a spectacular success. Well, okay, maybe. But you would think.
Yeah. During a pandemic, and then if anyone says, Dr. Bhargava creates great omelets, but now she's cooking for 10,000 of us. And did you know she's immune from being sued by any of us if we get food poisoning? Like, that's when it's like, all right, there's grounds to, at the very least, question it.
Right. Well, so let's start with the issue with mRNA, or why mRNA. So I guess traditional vaccines, which are for polio, chickenpox, mumps, measles, rubella, they are mostly viruses that cause the disease. They are taken, and then normally you grow them in chicken eggs. And so as you grow them from the first time to second time to 15th time, they now start thinking that chicken is their new host, and it takes time, and you have to go through that process so that it no longer is thinking of humans as its host, and it's thinking of chicken as its host. It's replicating there. And so it becomes weakened, if you like, because when you re-give it to humans, it's going to take that much amount of replication time before it starts to think of humans as its host. So in a way, if you didn't control that, that becomes a zoonotic transfer. Do you understand? So
Yes.
if those chicken eggs leaked or didn't get contained, you would have now a chicken epidemic or whatever.
Yeah.
So now you take that virus from grown in chicken eggs, and then you weaken it further by either chemically mixing it with something so that it kills it, or by heat inactivating it, and then you give it back to people. And in that case, obviously the route of infection is not the same, but it still does what a natural infection would do, because it's largely unmodified. And yes, it's not in the same route, and that's important, but nonetheless, the virus is there, and your immune system will make antibodies to different part of the virus, and you will get this polyclonal protection. Now, the problem with that is that the number of virus particles that can be grown in each egg are finite. And so you need a lot of chicken eggs, and you need a lot of time, right? So if it takes 5 days to grow in one cycle, you need 5 times 15, 75 days before you get the final product. And now it's too late, right? Because we are in this chaos, and people are dying. And if you don't do something, we are going to die tomorrow. We don't have 75 days to wait. So now we need something tomorrow. Except that even mRNA can't be made tomorrow, because that needs to be scaled. The raw materials for making mRNA are expensive. It's not just that you need one raw material. So you need you need a template. So what I did is, I'm going to try and explain. So let's just say that, RNA is ribonucleic acid, and normally there's a special kind of RNA that will make protein, and it's called mRNA, or message RNA, because it takes the message from DNA, or even from, if it's an RNA genome, from RNA, and makes it into another language, which is the language of protein. So to make mRNA, these mRNA vaccines in the lab, we have to make first a reverse copy in a DNA. So let's say this is your DNA, like a circle.
Okay.
In this case, it's whatever. It's and each of these beads is your your nucleotide. It's and so before it can be made into a message in the lab, this DNA, which is, we call it in a plasmid, it's normally circular, so I can't, but it has to be made linear. And that is done by an enzymatic cut, because the start site from where it's going to make RNA from this DNA template is here, and the end site is right behind it. So if it's not linear, it's going to go on making this round stuff. It doesn't know where to stop.
Okay.
And so it has to be made linear like this, so that it knows this is my start and this is my end, right? So now this is an enzymatic cut. When we do it in a test tube, we can't guarantee that 100% of the molecules will be cut. So let's just say, for simplicity, we put 100 of these plasmid molecules in a test tube, and when we put the enzyme, we don't know if all 100 will get cut. At best, maybe 90, and 10 will not.
Okay.
Now, let's say 90 are cut. Then it starts making this RNA, and it goes from one end, which we call, for directionality, the 5' end. It's because of the chemical structure, or the upstream end, to the downstream end, or the 3' end. So it'll make the enzyme RNA polymerase will bind here and start making this, copying each of those and making your RNA from this DNA template. Now, to make that, it needs raw material. So it needs this enzyme called RNA Pol2, which is what is in vivo, but in in test tubes, we don't add RNA polymerase II. We, from this DNA template, we add other enzymes called T3 or T7 or sp6 polymerases, which are of bacterial origin. Then they need the raw materials of RNTPs or ribonucleotides, and then they need magnesium, and then they need some other, obviously other stabilizing agents. But what happens is when you start scaling it up and you start adding too many of the ribonucleotides, there's a balance. They start binding to magnesium, and you take away or squelch or chelate magnesium, which means the enzyme now does not have magnesium available. And if that's the case, the enzyme is not going to work. So we have these scaling issues in the lab, and there's an optimum concentration or amount above which scaling is extremely difficult. So they would have to make millions and millions of these reactions before they can get the amount of doses that's needed to be given to people. And of course, you're setting up these independent reactions, which will vary, right? So we just, in one tube, you could have out of 100, maybe only 50 were cleaved. And so the other 50 will be making these round transcripts or messages. Now, that's not the only thing. Once the message is made, if it stays like this, and you give it in the cells, it's going to get degraded because this is not protected. So, so what it needs to do is it needs to then add, if now from that language, you've gotten another language, right? So now it needs to add at the 5' end something which is called a cap. So, and then on the other end, a tail, which is a poly A tail. And this is the hallmark of eukaryotic or mammalian RNA, so that this is now going to be made into protein. Without this cap and the tail, the RNA is not stable and it's not going to be recognized by our protein machinery and cannot be made into protein. So this is the other challenge. In vivo, in our cells, there's all this process that's going on simultaneously. Whereas when you're making this in a test tube from that template, which didn't have any information to add this 5' cap or a 3' poly A tail, so you have to add those ingredients in the test tube to add this capping and tailing. And that process itself is not 100% efficient either. So if you want to do it in one step, there's a 2-step process, and you can use enzymes which are much more effective, and they can be 100% effective in adding caps. But those are 2-step process, which means you have to, after you've made RNA, you have to purify, and then you have to add another set of enzymes and buffers and blah, blah, blah. So that takes time. So you take that shortcut and you do a 1-step process. And so as the mRNA is being made, it'll also add the cap and the tail. But that is only 60 to 80% efficient. So that means now, let's just say we got 100 mRNA molecules, only 60 to 80 of them will have the cap and the tail. The remaining 40 to 20, what are you going to do with them?
Yeah.
They're not going to be making any protein, but they count towards your microgram that's going to be given to people.
Got it.
Right? So now you've got that. So if you don't purify it, so now, let's say you purify it, and you purify it normally, and you have to purify it because you have to remove this DNA template from your mixture, because if you don't, and you give it to us, then this is going to make RNA in you.
Okay.
Right? Because we have the enzymes, or it can interfere. It may not make it in us because it may not have all the signatures, but it may cause issues. So you have to get rid of the DNA template, and you also have to get rid of all the other enzymes that were used to make the RNA and then used to cap and things like that. So you have to purify And you have to purify, so they normally use what is called size exclusion chromatography, because that's more faster. In the lab, when we do it, what we do is we actually precipitate, or
Not freeze, but
not freeze,
cool
but from the
down.
solution we precipitate it out, and the precipitation ensures that only RNA can precipitate, and along with DNA, but nucleotides or the raw materials and the enzymes don't precipitate. So we normally add like a salt, like a lithium salt or a sodium salt or an ammonium salt, and then you add alcohol and you precipitate it out, and then you wash it and you resuspend it, and that's what we use. But you can't do that for, this millions of batches that are going on. So they use normally some sort of chromatography to separate it, or a column or whatever. So if you then use, let's say I want to make sure that I have all the mRNA molecules that have the cap and the tail, then you can have a column that will make sure that it binds the caps and the tails. And so anything that cannot bind or didn't have that will just go through.
Okay.
But that's a very laborious and expensive process, and you may end up breaking the tail because it's fragile. You may end up. So they use size exclusion. When you start doing size, it's not like it's a precise size. It's there's a very small difference between the mRNAs that get capped and tailed in size compared to that don't get, and the original template is also of the same size. So all of them will now be in this mush, and that's what you're going to get. So yes, you may have excluded the proteins, the enzymes, and the raw materials to a large degree, but not completely. Now, all this is being done in a machine or whatever, right? So this is where I think the issue happened with 1.6 million doses of Moderna vaccine in Japan, where the machine introduced, I guess, some steel particles in while packing, or maybe while aliquoting it into those vials. How they were missed, who knows? They're not like microscopic or whatever. They were fairly big metallic particles, and now given to people, so people died, obviously, because they're going to get lodged in your, and maybe they were, if they were not given in the muscle, it went into their veins. So these 4 people died. And so Moderna recalled all 1.6 million doses. But we're not even in that talking about all this gamush that's there, right? And so the question is, you have all of this, you have to package this into, this is not going to be taken up by your cells. So you're going to have to package this inside these, what they're calling lipid nanoparticles. So remember, like I told you last time, the lipid nanoparticles are like, imagine bubbles, right? So you take when you blow bubbles, you take that little stick and you start blowing bubbles. Now the circle from which you're going to blow that bubble is the same size, but the bubbles that you get are of different sizes. So you can take the lipid and you mix it with this RNA, and the RNA is going to get encased or encapsulated in this lipid thing, or lipid nanoparticle. The problem is that this lipid nanoparticle is not the same size, right? So you can have this size, or you can have this size, and in that there'll be more of these.
Okay.
Right? So if it's one size, then that's great. You know exactly every lipid nanoparticle has only 100 molecules, but because they are different sizes, some can have 100, some can have 200, some can have 50, some can have 10.
Yes.
Big variability. Now you take all of those different sizes and you put it in a vial, right? That's the vial. And now it has to be, it's frozen. So when you bring it down to room temperature, or you thaw it, normally, again, when we want to break open a cell, which is also made up of its outer layer is a lipid bilayer, we do this freeze-thaw process, which is you freeze it and then you shock it and you thaw it and it breaks open. So yes, to prevent that from breaking open, they mix sucrose or some sort of sugar so that that acts as a cushion, and so you don't rupture it open. But nonetheless, let's just say, even some of them open up, then you've lost that particular amount because that's not going to be taken up by your cells. The other issue with that is that once you thaw it, now you have to dilute it before you give it to people. So you're diluting it 1:3. The assumption is that 1/3, 1/3, 1/3 will all have, whatever, 30 nanograms or 50 nanograms, which Pfizer or Moderna, whichever dose you're looking at. But that's not true because the size distribution you cannot control in that suspension, right? So in that 1/3, you could get everything.
Mm-hmm.
Or you could get, instead of 30, you could get 50, or you could get 60. And the next person may get only 10, and the other person may get nothing or very little. So you've already created that dose discrepancy. Now, the issue with that is that if a person got, instead of 30, 50 micrograms, it may, and we are not really accounting body weight or any of that into taking that into consideration, right? So, or it could have sat out for a longer time by the time the third person got it, or whatever it is. There's so many issues, or they took it out from the freezer, put it on ice to thaw, and then, the time variabilities between one site versus the other site, none of that can be controlled. You saw a lot of people giving these injections without wearing gloves. RNA is extremely labile. The other issue that was pointed out by the people who were working on this Moderna and Pfizer, the challenges that they were facing is that this mRNA that we make, normally the nucleotides that it has, they are very susceptible to degradation. So to prevent that, they use these modified nucleotides. So instead of, if let's say normally it would be only there are 4 nucleotides, so 4 different types, but one of them, which is uracil, is replaced, and now you get this pseudo-uracil, which makes the mRNA more stable. Now, they have documented, Moderna and Pfizer and BioNTech, that modified RNA actually stays around for a long time. So what happens is normally each mRNA has a particular, what we call half-life. That means it'll stay in the cell for a certain number of hours or days, depending on what its half-life is, and after that, it's degraded. But once you modify it, we don't know what the half-life is. They never tested it. So, but what they did find is that modified mRNA stays around for weeks and weeks. So at this point, we do not know for how long that mRNA is staying in us and where all it goes, because it's being given in the muscle, and your immune cells take it and then they circulate, and so you don't know. And then so they never did the biodistribution study. The other issue is that you could have in this mixture mRNAs that are not capped or poly A tailed, so they're not going to be making any protein, but they are there now. So, and if they are stable and not getting degraded, what are they doing? They're not making protein, they're hanging around. Are they, but because they're non-self, my prediction is that they're evoking an immune response, which is different than what is predicted. the third issue is that these mRNA, because they're hanging around and they're non-self, we have certain enzymes called DNA theta polymerases, which are very good at taking these non-self DNA and RNA and integrating it into our genome, because just like CRISPR, which is an immune system for bacteria, which works by recognizing the virus that's going to invade it or pathogen that's going to invade it by comparing it to the sequence it had integrated in its genome from using CRISPR before. Probably like that, we have fairly large amount of our genome, 40% or more, has these viral signatures in us, which are called line and sign elements. So maybe it's integrated so that in future viruses that come in, we can see that we've had them before and they are there for whatever purpose that we don't fully understand. So this integration can happen in places where the genome has key function. And if that happens, then who knows what the outcome will be for that particular person. So there are so many steps that we don't understand, that we haven't studied. And unfortunately, just by saying that we have an emergency or declaring it doesn't mean that you can bypass all this. And none of the studies that were done before this was deployed actually even address it. So you would be surprised to know that even in animal studies, they had never done repeat dosing. So here we are repeatedly dosing, and when they did in the few studies that were done, I don't remember by which company, they found that the lipid nanoparticle redosing causes toxicity. It's toxic. So it's just amazing that we knew so little. Even though the technology has potential, there are a lot of issues that we haven't worked out that need to be worked out before it can be deployed, especially in healthy people. Now, when you think about somebody who is got a chronic disease where the quality of life is really bad, they're not functioning anywhere near what a normal human being is functioning. And if you then take a therapeutic which is not tested, let's say like this, and give it to them, and they show improvement in quality of life and things like that, then the benefits are so much more than the risk and adverse events that they may have. That and it's confined to a subset of people, but vaccines are different. We give them to healthy people. So that's your, in terms of just trying to touch the surface of this iceberg of what can be the issues in mRNA production and this batch effects, or whatever you may call.
So all of these excuse me, all of these points in the production of it that may cause uneven distributions, you maybe aren't too worried about. If you have a comorbidity, if you're older, you might look at it and go like, yeah, like it's fine. It's, if you're in a car crash going 80 miles an hour, yeah, the airbag isn't 100% safe, but you kind of don't have any other option. So it's kind of the best thing for it. But you don't want to apply that to everyone. So if you don't know how many the plasmids are cleaved, and then you don't know, so if that's anywhere from 60 to 80 to 100%, and then if you have the actual blowing of the bubbles, and then you don't know, so you already have the 60 to 80 to 100%, that makes up X mass. And if you then take up X mass and know that you have to dilute it 3 times, you're blowing the bubbles. You're already taking something that is anywhere from 60% to 100% of what you need. And now you're dividing that into three separate, well, in this example, three separate, bubbles. And you don't know how much is going into each of those. And then once it gets into the cell or into the individual, we don't have long-term studies. We don't know what's going to happen to each person. And then also tied in is the pseudo uracil or pseudo uridine. We don't know how long that's in there. So there are these multiple moments of uncertainty that I'm terrible at math, but they multiply, right? If I want to, whatever, if I take a deck of cards and, what are the chances of me pulling out an ace? Well, it's 4 and 52. Okay, but what if I take that deck of cards, shuffle it, and then pull it out again? Well, it's 4 out of 52 times 4 out of 52. So your chances of certainty go lower and lower and lower. By having multiple moments of that uncertainty being duplicated, and then the wild card of you're ramping up production for the first time ever of a new technology during a pandemic. And then not even including the fact that you might get steel particles. All of those lapses might be acceptable if this was a modified Ebola. You might look at that and go, it's not perfect.
Well, I'm going to interrupt you there and say, no, I don't think it's acceptable under any condition.
Okay.
And I don't think so. To add to all of that is, of course, you're giving that mRNA, but it's our cells ultimately that have to make the protein. So you don't know what's the efficiency there. And it depends where it gets to. So maybe your heart cells are more effective in making the protein compared to your liver cells or whatever. I'm just, but I just want to correct that the example that you gave about giving it to people with comorbidities or immunosuppression, that no, it's not acceptable. What I was talking about is, let's just say you have a terminal disease like cancer or something like that. It's different because you don't have, let's say, hope, or your quality of life is so bad.
Gotcha.
Then you might be willing to try something that's radical, where or just because you're desperate. But this is not a desperate scenario. Most healthy people, we know that 80 to 90% of the people didn't really have severe disease, right? Oh, and even with the numbers that we have right now, 2% of the people died. Yes, 2% globally is a big number. And if we say that's not acceptable, why is it acceptable for people to die in wars? Wars are for choice. This is not a choice. We send our healthiest, youngest people, train them, fittest people, and send them out to die. That's acceptable? How's that acceptable? We there are so many things that are just not right. I mean, why is it acceptable just because freedom, and literally, like, I don't want to get into things that are political, but there are a lot of things that don't make sense, and but we do it. Assault weapons, why are they freely available? It's not about your right. What about my right to live? Right? Sure. Because you could have somebody who's unstable or whatever. So you have to have some checks and balances, and it's so this without testing this, without having a technology that you can feel confident about, it's a disservice you're doing to the society and to science. And it's and then people who are raising legitimate concerns, who understand this, like me, I would not call myself an expert just because every time I go and um, you know look at certain things or look at my data, I learn more new things. And so there's still so much to learn and still so much to discover that I think you remain a student all your life in not just in science, I think in any field, right? There's just so much going on. And so but in some ways, when we work like that, we have to wear blinders, right? So if we are just think about what's happening in a cell, if you're looking for a particular protein, and let's just say that protein is like a red color Tesla car. So now, but you have to imagine, not just LA traffic, but all of US traffic is in your, because that's the number of proteins that are going around. And you have to only, you can only focus on red colored Teslas, but it doesn't mean that the other cars don't exist.
You're not in a vacuum.
Those exist, and they can influence or change how the red colored Teslas will drive or where they may end up or whatever. But if you only just you discount everything else and only focus on those and the outcomes there, you can reach very wrong conclusions just because you're only looking at how one red color Tesla impacts the other red color. You forgot that in between there could have been a Toyota or another truck or something else that could have then influenced how this red Tesla behaved with the other red Tesla. So that's how, things end up happening, and that's what they're doing, and cherry picking information, right? So when we do this in vitro or even in animals, things that don't work out, normally they're ignored, and they're ignored or they are discarded, or they say, oh, I don't understand, instead of saying, or if nothing happened, that's like a negative data and you don't report it. So there's a lot of gaps that we have. It's not a lot of time there's no control done, right? So there's no control here to say, is this really effective? What if we just took some other viruses mRNA and put it in? Do we get similar kind of protection just because it's non-self? Because if you are only counting it by measuring antibodies are made to an epitope, meaning this, let's say from this RNA, you get, I'm going to pick up another one, a different kind of this is a protein, right? And the protein gets folded. So as in when it gets folded, the surface of the protein that's exposed is what the antibody is going to get made to. So that's called an epitope. The folding of this protein is dependent on the speed by which this mRNA is being made into the protein as each amino acid is added. And that depends on the environment. So for example, the fold and other proteins that are there. So for example, if it's being made in your heart cell, it may fold differently than if it's made in your liver cell. And so the epitope that may get exposed or the shape may be different. So the antibodies are not going to be the same as, and yet we are comparing that to natural infection versus the vaccine antibody. So that itself is very flawed. And the second is that when you get a natural infection, the thing that neutralizes those neutralizing antibodies, or IgA, are not made to spike protein. They're made to other parts of the virus. So this, whereas when the studies that most people are talking about, oh, you have a high antibody titer, they're looking at non-neutralizing antibodies like IgG or IgMs, and then they just say it. It's like just saying it doesn't matter. They're just people. And then if you have people, you have protection. They're protecting your house. But all people are not the same. What if, if some people were trained soldiers or whatever, they will protect it much more, even if the numbers were not the same as the untrained people compared to, so you can't just compare antibody levels and just say, oh, that's it. You have to see what kind of antibodies, and none of that is being talked about. It's all lost in this noise and the chaos that's been created. So I think I don't have anything more to add. We are coming up to an hour.
No, it's well, two things is people are talking about that. The difference is someone like you or someone like Dr. Malone, who you guys are humble and you won't call yourselves experts, but compared to the rest of us, sure, yeah, and any person that's truly learning anything knows that they're a lifelong student. That's a given. But for all intents and purposes, you guys are experts. You guys are the individuals saying there are too many. You're the ones taking time out of your day to explain to someone like me and my listeners with some beads about what is going on and the dangers in it. You're not getting paid for this. I know I certainly am not. These are people that care about what's going on and they want to inform you. Most of that, 99% of what you told me today, I didn't know. Now, if you are someone that knows all of that and you have malicious intent to make money, to corner the market, those are the people that are going to simplify it down to, you need a couple of these. It's like recharging a battery. It's like proteins defending your house. It's like soldiers. It's all good. Don't worry about it. And then if you do seek out, well, what are opposing voices say? And then those are all squashed. That's where the problem is you are trying to explain it to people and you can't even find that. And to kind of backtrack to what I said earlier about it being acceptable, I was using that as an example. I personally don't believe it's ever acceptable to force anything on anyone ever. That's my own personal, it's my choice. Leave me alone. Get out of my house. Leave me alone. It's not acceptable. It is always a choice I want to make. If I want to sign up to get on one of Elon Musk's rockets and go to Mars, I might blow up on the launch pad. I might die in space. I might die on impact. That's my choice. I'm an adult. I'm 32. If I want to die on Mars, that's up to me. But it's not something that you can force people to do. And this is, again, that's where my decision ends. But let's play with the idea of, if you were be allowed to force this, you can't shut down all criticism. You can't give legal immunity and financial immunity to the companies. You can't have a widespread, coordinated suppression of all dissenting voices, especially when it's not an emergency. And as you said, and as I agree, not even for Ebola, especially not for something where the vast majority of us. I've had COVID twice. It sucked. And then I got over it. I think the last time you and I spoke, I had COVID, and I told you, I was like, I'm still going to the gym. And you were like, don't do that. But like, you're fine. That is where all the problems lie with this, is even in the absurd thought experiment exceptions, it's Ebola and it's during a war, which still is not acceptable. We're not even there, though. It's for a virus with a 99 point whatever percent survival rate, and you're quashing all criticism. And it is a patented technology where these companies are printing money, and they are and there is, they're in bed with government paying for advertisements. And like you said, you don't want to get in political, but like, that is unfortunate. It's a pandemic touching every person on earth. You can't avoid much like the red Tesla cars and all the traffic of the United States. You can't just say we're only going to focus on the red Tesla cars. There are a lot of other cars out there, much more. In that same way, we can't focus in on just the vaccine and go, I don't want to talk about the politics of left or right or America versus Russia or China and Mexico. Good luck. You can't separate them. That's an unfortunate fact. And really what it all comes down to is you have to boil it down to the most fundamental things and say, if this is going to be so complex and touch so many aspects of life on earth, you have to have some ground rules, and it's your right to say no, and your right to discuss it. And when those are shut down, that's when it's not good. No time in human history, ever, not once, have the people censoring and shutting down scientific debate been on the right side. Never once. I'm sure there were a lot of people 500 years ago that were saying they just need to shut Galileo up. He's spreading misinformation. Copernicus is up there thinking that we revolve around the sun, and he's spreading harmful disinformation. Yeah, it eventually you will be proven wrong. And the difference is no one died because of that. Well, I know they killed the astronomers. No one died because of that. Now people are dying. Now kids are getting myocarditis. There's a lot wrong with this. But I know you're muted. I can't hear you.
No, I've muted myself
Oh,
because
okay.
just
Sorry. Yo, you were talking about.
I have to go in 5 minutes,
Yep.
but I think, I mean, it's not a discussion that we can have, but in terms of the what led to this stage we are at. But in short, we are here because I think in the past, every time they tried to get a vaccine going, let's say for the SARS or MERS or Ebola, the pandemic or the epidemic died by itself. So billions of dollars were spent in developing those vaccines. They could never be deployed. And now we have, so much of loss, and the pandemic went away. So what do you do? So this time around, they had to make sure the pandemic stayed and you could deploy it even though it was not needed. Because if they didn't, we would be in the same stage. And literally, Moderna and BioNTech were dying. As of August of 2019, they were dying. And suddenly, they didn't have anything that was working for them. And suddenly, in 6 months, they are a spectacular success. Doesn't happen. You have to question that. And it never can, when something is too good to be true, it's too good to be true.
Yes.
But the just one point that I want to make is that the issue that we deal with these days is the difference between people like me who were trained old school way and Dr. Malone and others. There's still in the last couple of decades, there has been a lot more advancement in technology, in development of kits and things like that for experiments that we do in the lab. So when I was growing up doing my PhD, we didn't have kits. We had to make all the solutions. So we know what goes into each of those, what's the process and things like that, and we can troubleshoot. A lot of my students can't. You buy the kit off the shelf, you just mix one thing with the other, and that's it. And if they sometimes run out of something, they don't even have the know-how to look up, and sometimes that missing ingredient could be something like 100 millimolar sodium chloride, and they are like, oh, we need to wait before it comes. Why do you need to wait? Can you look up what is the thing? You can just make it. They don't have that know-how. So most people actually don't have that know-how. So they don't really know. It's like, oh, we have all this, it goes into this black box and you get this out, right? So they don't know how to troubleshoot. They don't know what we're going to get out of it. So they don't understand. A lot of people don't understand. And the other thing that's happened is this siloing of the areas, right? So if you are a neuroscientist, you only study certain aspects of brain. Even in there, it's, whatever. You're an immunologist, you're studying only the immune system, but the immune system doesn't work in isolation. It's working in coordination. It's there to protect your other organs. If you are a renal fellow, you're only studying kidney, but the kidney doesn't work again in isolation. It's doing because of what else is going on in your body. So that's the other downside of people these days, which they don't just have this cross pollination, if you like. And I feel very fortunate that I do have that, and I've worked in various fields. So if you, like I've said before, if each of those areas is a different language, it's like, if you're speaking English and Italian and Spanish and you can crosstalk so you have a much more richer life. Similarly, I feel fortunate that I am able to understand molecular and cell biology, and I can understand physiology, and I understand immunology because I study stress responses, and there is no part of your body that stands as a bystander and say, oh, you deal with this stressor and I'm going and resting today. I'll come back to it another time. No, it doesn't happen. And I realized that just working with the brain to look at stress responses is incomplete. So I've had to learn a lot and cross-pollinate. And so I think I'm fortunate. So with that, I have to go, but it was fun talking to you. And if there are any questions, we can, take them later.
Okay, Dr. Bhargava, thank you so much. You're absolutely brilliant. Thank you for the free science lesson. It's been 10 years since I took biochemistry, so I think I had a little awaken some old demons, remembering caps and tails and plasmids and 5 and 3 and ATTGA and whatever. So that was bad, but thank you so much. Thank you for your time. Thank you for the wonderful lesson. And yeah, thank you for hanging out with me for an hour. I will
Okay,
put
talk soon.
Yes, ma'am. I will send it to you when it's uploaded, and hope you have a wonderful rest of your day. Thank you so much,
Yeah.
Dr.
Thank you.
Bhargava.